Hello everyone, welcome to the ITS Academy. Today we are going to discuss the topic that is your detection of sulfite reducing anaerobe, that is your Clostridia. Okay, we all know that this Clostridia is what? It is a microorganism, or in microorganism it is a bacteria. And the sources of this Clostridia are soil, water, and intestine of intestinal tract of human, dust, feces, and silage. If it is bacteria, that means that it is come under the category of harmful bacteria. That means that it can cause a disease to the human being. Okay. So here our sample is what? Our sample is water. And we have to detect this Clostridia concentration into water. And it is very important because it can cause a serious infection into the human being. Serious infection to the human being. Okay. And also cause a infection into our animals. And this infection included botulism, tetanus, gas gangrene, and antibiotic associated clotis. If it is earlier detected, then the prevention become easy, treatment become easy. That's why it is very important to detect this sulfite reducing anaerobe, that is Clostridia, into the water sample. Now start our lecture. Introduction. We all know that the sulfite reducing anaerobe, that is your Clostridia, they are what? They are relatively large gram positive. Gram positive means that when we perform a gram staining, then it give a purple color colony during the microscopic visualization. Okay, they are rod shape and they are anaerobic bacteria. That means that they can survive even in the absence of oxygen. Okay, that is the quality of the Clostridia. Now, this is the introduction part. Now, come to the next slide. Principle. Detection of the sulfite reducing anaerobe by inoculating the 50 ml of sample into the equal volume of double strength of differential reinforced Clostridium medium. Okay, here the culture media is what? Culture media is a DRCM. Okay, so what we do for the detection of this Clostridia, that is your sulfide reducing anaerobe, we inoculate the 50 ml of sample into the equal volume of double strength differential reinforced Clostridium medium. That means that we take 50 ml of media also. Okay, and which is followed by an anaerobic incubation at 37 degrees Celsius for 44 hours plus minus, which give a result of reduction of sulfide and the precipitation of iron second sulfide in a medium, developing a black color in a medium. Okay, if the black color occur inside the medium, that means that they give a positive result of Clostridia. Okay, that means that it give a positive. Sample is considered as a positive. Now the next is your culture media. So we all know that we use a reinforced Clostridia medium DRCM. So the composition of this media is we have a yeast extract. Now this composition is for a 1 liter or 1000 ml distilled water. Okay, for this we have a yeast extract, we have a peptone, glucose, soluble starch, then sodium chloride, sodium acetate, cysteine hydrochloride, agar. Now we required yeast extract 13 gram, peptone 10 gram, glucose 5 gram, soluble starch 1 gram, sodium chloride 5 gram, sodium acetate 3 gram, cysteine hydrochloride 0.5 gram, and agar 0.5 gram. Okay, these all the ingredients first dissolve into a 500 ml of distilled water. When it is completely dissolved, then the remaining 500 ml of distilled water will be added and make up volume 1000 ml. Then after that, we have to maintain its pH 6.8 plus minus 0.2. Then we do a autoclave. Okay, autoclave will be done at 121 degrees Celsius, 15 psi pressure for 30 minutes so that all the contamination will remove, or it will eliminate the microorganism or reduce the concentration, reduce the number of the microorganism. So this composition is for a 1000 ml. If you have to prepare 500 or 100 or 250 ml or more than 1000 ml, so you have to calculate this value according to your composition of the distilled water. Now the next is your procedure. Aseptically clean the surface of the bottle or water pouch cup which contain a sample with the 70% of ethanol. Suppose that in this bottle we have a water sample. Okay, so it should be cleaned by a 70% of the ethanol with aseptically, so that if there any contamination, then it will be reduces. Now we have take a aseptically 100 ml of distilled water into a sterile water and heat it at 75 plus minus 5 degrees Celsius for 15 minutes. Now from this sample bottle we take a 100 ml, and this 100 ml will be taken by a aseptically withdrawal and put it into a sterile bottle. A sterile bottle means that the bottle are free from microorganism. Okay, then this bottle are heated at 75 plus minus 5 degrees Celsius for 15 minutes. Add 50 ml of the sample after the heat shock to the 100 ml of bottle, in which 50 ml of double strength DRCM are present. Okay, so what we do, we have a bottle in which we prepare our we have this DRCM. So we add 50 ml of sample after the heat shock at that bottle in which the media are also present. Cap the bottle tightly and incubate under a anaerobic condition. That put it inside the incubator at 37 degrees Celsius for 44 hours. Iron wire heated to redness can be placed in the medium before inoculation so that it will enhance the anaerobic condition. Observe the water for blackening. So this is a end point when a black color are observed, then it means that the Clostridia are presence. Okay, this is the observation. Now the expression of the result test for the sulfide reducing anaerobe Clostridia is equal to present or absent per ml per x ml of sample. Suppose that we have a 220, then for 100 ml. This is a presence or absence. This is how we express our result. So this is all about the detection of the sulfite reducing anaerobe, that is Clostridia. If you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.